Assalamu alaikum. Hello everyone. Today we will be doing the lecture 2 under the topic of cells, and we are still on unit 1.1, in which on the screen you can see we had covered the first two learning outcomes which have been highlighted in green, and today we will be covering the third learning outcome, which is identifying on diagrams, photomicrographs or electron micrographs, the ribosomes, mitochondria, nucleus, cytoplasm, and cell membrane in an animal cell. So let's start. Again, I already told you before as well, I always share this document, one of the pages in the document from the scheme of work, which is basically used by teachers. But if any student is interested, he can go through the details of how this learning outcome is actually covered during the class. So as you can see, I have highlighted this. So again, in the scheme of work also, you can see that today we will be focusing on identification on the diagrams, on photomicrographs, which means the micrographs that have been obtained by taking a picture through the light microscope, the electron micrographs, to visualize or to observe the structures, ribosomes, mitochondria, nucleus, cytoplasm, and cell membrane in an animal cell. Let's move on. As you remember, in the last class, I had drawn these two diagrams in front of you to give you a basic idea of how an animal cell and a plant cell looks like. Again, focusing on the same point once again, there is no such thing as a typical animal cell or a typical plant cell as far as the shape or the structure is concerned. Yes, there are some distinctive features which are found in the animal cells and some which are found in the plant cells. But the animal or plant cells, depending upon the kind of the function that they can have any shape or any size. So we'll be focusing on the various features of the animal cell. On this drawing also, you can see, I'll be using my highlighter, that this structure lying in the middle of the cell, which controls all the activity of any living cell, is the nucleus. The structure that surrounds all the contents of a cell is the cell membrane. It's a delicate structure, it's a living structure, and it controls the entry and the exit of various substances into the cell. It protects the cell. It's a partially permeable structure, which means it allows certain substances to pass through and certain substances not to pass through, hence the name partially permeable. And then there was a jelly-like substance which is found in the space that lies between the nucleus and the cell membrane, which is known as the cytoplasm. Cytoplasm is, again, just to recap, the place where all the different chemical reactions take place, right? And then besides the cell membrane, nucleus, and the cytoplasm, today I will be showing you photomicrographs and electron micrographs, where we will be identifying two other structures found in the animal cells, which are known as the ribosomes and the mitochondria. Just to give you an idea about one terminology which is known as organelle. I'll be using this word organelle from now on instead of using the word structures. So within one smallest structural and fundamental unit of a living organism, which we call a cell, we have certain other smaller structures like a nucleus or small vacuoles, or today, which we will be doing, ribosomes, even smaller than vacuoles, or mitochondria. So all these different structures which are found within a cell are a small structural and functional unit within themselves. We won't be calling them cells, but we'll be calling them organelles. Why? Because an organelle is a smaller structure found within the cell which can work on its own, and it has its own specialized structure and function. So I would say the nucleus is also an organelle. The mitochondria is also an organelle, the vacuoles are also organelles, and the ribosomes are also organelles. So now I will be using this word organelle for all these different structures found within an animal cells. So let me show you some diagram. I'll be zooming into this diagram, which I had shown you in my last class also. So this is a light micrograph of a human cheek cell. Why am I calling it a light micrograph? Because it has been obtained by taking a picture through a camera, uh, which uses light energy in order to look at these cheek cells with some magnified lenses, right? So hence the name light microscope. A very good quality light microscope can give you a total magnification of 1500 times. And here you can see that this magnification that you can see is around 400 times. Okay. So this diagram has been taken, or it has been magnified by 400 times. It shows you the nucleus, which is very obvious, this one. It shows you the cell membrane. Okay. And it also shows you the space between the cell membrane and the nucleus, which is the cytoplasm. Yes, it does show you that the cytoplasm is granular, but we can't see the ribosomes or the mitochondria or the vacuoles so clearly. Why? Because it is only magnified to 400 times. Had this picture been magnified to 1500 times, or maybe had this picture been taken by an electron microscope, we would have been able to see the organelles more clearly. So this means that this kind of a magnification has a very limited resolution, right? But still we can see cell membrane, nucleus, and the cell membrane, nucleus, and the cytoplasm. Now let's look at another diagram. Now this is not a light micrograph. This one is not a light micrograph, but it's a handmade drawing of how a cell would look like if it is seen with a light microscope of a higher magnification. Let's assume that this picture was taken from a light microscope, a very powerful light microscope, let's say, which gives you a magnification of 1500 times, or maybe if not 1500, let's say 1000 times. Then, and if I make a handmade drawing of such an animal cell, it will enable me to look at certain smaller organelles very clearly. As you can see, we can see the nucleus more clearly over here. Okay, we can see that the nucleus is bounded by a very clear boundary, the nuclear membrane. Within the nucleus, we can see the nucleolus also, right? And we can also see these red colored organelles, which are known as the mitochondria, right? So we can see the mitochondria, we can see the nucleus and the nuclear membrane and nucleolus more clearly. We can see the cytoplasm also more clearly. And some small structures have been drawn over here, but they are very difficult to identify. So this means that we would be needing even a higher power magnification in order to observe structures which are as small as ribosomes. In fact, ribosomes are those organelles which cannot be seen under a light microscope. In order to visualize these structures, we will be needing an electron microscope. So let me show you that picture. Now, this is an electron micrograph of a liver cell, and which has been magnified to around, now you can see 20,000 times, so much more than the magnification of a light microscope. It's an electron micrograph. It shows you even finer details of a nucleus, the nucleolus, the nuclear membrane. It can show you cytoplasm, and it can also show you mitochondria. I'm just encircling the mitochondria so that they become visible to you. So lots of mitochondria. Mitochondria, just to give you an idea about what do they do in our body, these are organelles found within the cells, which are the site of production of energy, which means that these are those organelles where energy is produced. So if we had no mitochondria, we would have no energy to function, because for every activity that we do, we need lots of energy. So this energy production takes place inside the mitochondria. In some books, they are also known as the powerhouse of a cell. So just like a substation where electricity is generated, the mitochondria are the powerhouse of our cells. We can't see the ribosomes over here. So to visualize them, I'll show you another diagram. Yes. So over here, this is as you can see, even a higher power magnification than the previous diagram. It is an electron micrograph of a cell which has been magnified to 30,000 times, where we can see these tiny ribosomes. Now, although they are not so clear over here, but I'll just color them in red. Just imagine that they're not so clear over here. So I'm drawing them here and there. These tiny structures which are found here and there scattered, okay? And they are known as ribosomes. Why are ribosomes so important? Ribosomes are the site of protein synthesis, okay? These are places where protein synthesis takes place. So ribosomes are the site of protein synthesis. So had there been no ribosomes, there would have been no protein synthesis. Proteins are an essential nutrient of our diet and they are needed for our body to grow. So hence protein synthesis is very important. Our muscles are proteins. Many structures inside our body like enzymes are proteins. The hormones circulating in our blood are proteins. Our hemoglobin is protein. So if there are no proteins, we would have none of these essential structures needed for life. Another very high power magnification, 30,000 times magnified through an electron microscope of a close-up of a mitochondrion. So you can see, the more we keep on increasing the power of magnification of an electron microscope, the more details of an organelle we can see, right? So this is again a high resolution picture of a mitochondrion, which can be seen with an electron microscope, which has been magnified to 30,000 times. So with this, we end with today's lesson in which we have been able to identify these structures on the light micrograph, on the electron micrograph, and on the drawings. So what were those four structures? Let's just go back and revise. Those four structures were cell membrane, nucleus, mitochondrion, and ribosomes. Cell membrane and nucleus and cytoplasm can be easily seen on the light micrograph, which is magnified to roughly 400 times. Mitochondrion can be seen on a high power light micrograph, whereas ribosomes can only be seen on the electron micrograph. So I end my lesson for today. Allah Hafiz.